So good afternoon. Welcome all to today's MENDEL lecture. It is my absolute pleasure to be introducing today's speaker, Professor Andy Myers from the Department of Chemistry and Chemical Biology at Harvard. I'll start simple by saying that Andy is an organic chemist, so don't go away. He has received his undergraduate training at MIT, working with Bill Rausch, and then proceeded to his PhD studies at Harvard under the mentorship of E.J. Corey. His independent career started at Caltech, where he, from the very beginning, pursued some of the most intricate molecules, natural products, called ene-diynes. These are very reactive molecules and DNA-damaging molecules. His group has proposed the mechanism of action for these molecules, studied it subsequently, and managed to prepare several members from this family. For the chemists in the audience, I want to mention that the pseudoephedrine asymmetric alkylation methodology is what started at Caltech as well. And these early achievements didn't go by unnoticed, and Andy soon found himself back at Harvard, now as a professor of chemistry. His research has been always intertwined with natural products that are complex and that are of biologically significant with potential impact on medicine. His group was able to recreate in the laboratory some of the most complicated molecules, but did so often in a concise and efficient manner. The idea of convergency as compared to linear synthesis that has been treated theoretically on numerous occasions really becomes alive through Andy's work. And I think this approach allowed him to not only make the molecules themselves, but also study their mechanism of action and develop novel analogues with improved properties. And this is particularly true for antibiotics. The research papers that came out of Andy's lab are, in my opinion, set a standard for scientific writing and scientific rigor. And Andy has been also a caring mentor of numerous students. Many of them went on to have successful careers in industry and in academia. For all of his accomplishments, Andy was recognized by numerous awards, and I hope he forgives me for not listing them out here. But I wonder if ultimately most rewarding will be his current dedicated pursuit of impacting human medicine with natural product chemistry. And so that will be for the introduction. I have one more obligation to do. MENDEL lectures have the tradition of getting some of the best speakers from life sciences to Brno, and tonight is no different. And so it is my pleasure on the behalf of the organizer of MENDEL lecture and the scientific community at Masaryk University to present you with a medal of Gregor Mendel.
Wow, thank you. Thank you very much. Thank you, Jakub. That was an incredibly kind introduction, and thanks to the organizers, anyone who had something to do with this. This is a very humbling experience. I'm extremely grateful to be here, and want to spend the next hour sharing with you efforts in my laboratory to bring new antibiotics to humankind. It's a subject that I've grown to be quite passionate about over the last few decades. Is the amplification okay? Is it a little too loud, perhaps? It's good? Okay. So I want to spend a few minutes talking about the antibiotics crisis in the world today. This is a highly literate audience. I'm sure you see in the papers and on television that there's great concern about the lack of effective medicines. Some numbers, I think, help to bring the problem into focus. In the United States today, more people will die from infections that they acquire during hospitalization then will die from AIDS, breast cancer, and auto accidents combined. Conservatively, that number is about 25,000 people, but it can rise as high as 100,000. The discrepancy has to do with what killed a patient. Did they die from cancer, or did they die from the bacterial infection that they acquired during cancer therapy? Conservatively, 20,000. The costs are staggering, and globally, it's estimated that nearly 1 million people will die from drug-resistant infections, about a third of those from tuberculosis. Very recently, the United Kingdom economist Jim O'Neill was tasked with heading a committee that spent, I think, a few years to evaluate the problem, to look forward into the future. And they published an estimate that by 2015, more people will die from bacterial infections that are resistant to antibiotics than will die from cancer, and the costs from this are estimated to be in the trillions. I find that it helps to talk about a specific superbug. This one is Klebsiella pneumoniae, and it is Klebsiella that has a gene. It's a carbapenemase. It's located on a plasmid, a little circular piece of DNA that it excretes into the environment where it can be acquired by other species of bacteria. And so as a result of this gene, this Klebsiella is resistant to carbapenems. I'll introduce the carbapenems in the next slide, but they are frontline therapy, IV antibiotics, the sort of drug that you would get in the United States if you were presented with a gram-negative infection. The first incidence of this bacterium was reported in North Carolina in 1996. Before it was not known. Within 3 years, that bacterium had jumped and into New York City, and every major hospital in New York City was reporting incidents of that infection. And by 2012, virtually the entire continental United States had reported examples of patients presenting with this infection. It's an extremely lethal infection. The mortality exceeds 50%. And there was a famous case in the United States at an NIH research hospital where a woman in 2011, she was 43 years old, she had a rare lung disease, and on top of that, they knew that she was colonized by this Klebsiella, this drug-resistant Klebsiella. So they transferred her from another hospital, and they put her in an isolation ward with strict gloves and gowns procedures. She was never in a room with another patient, and she only left that isolation room twice for 2 24-hour periods when she was in the intensive care unit. She recovered from her infection and was discharged after 5 weeks. But about 3 weeks later, a second patient, a 34-year-old male cancer patient, came down with a bacterium that could be traced through whole genome sequencing to patient 1. And this patient subsequently died from the Klebsiella. And before this went on for weeks, if not months, before the last patient was identified. And in all, 18 patients were infected with this bacterium, and 11 of them died. 6 of them could be conclusively traced to Klebsiella. Through whole genome sequencing, they learned that the patient 1 had transmitted the bacterium on 3 separate occasions from 3 different body fluids, and one of the transmission events involved a ventilator that had been sterilized, but obviously not effectively sterilized. So these are the sorts of infections that are out there, and you're going to see this Klebsiella pneumoniae, CRE, carbapenem-resistant Enterobacteriaceae, a few times in this lecture. So these are carbapenems, ertapenem and meropenem, frontline antibiotics which which many bacteria have acquired resistance to. I'll tell you now that all carbapenem antibiotics are fully synthetic. Not many people know that, even infectious disease specialists. So that means that these are built from scratch from industrial chemicals at a plant at companies like Merck and others. All third-generation cephalosporins, all cephalosporins are semi-synthetic. They're made from natural products, and I'll talk about that. And this is colistin. Colistin is, in many ways, an awful antibiotic, but it is the last line of resistance right now. There are many bacteria that are resistant to all of these and are currently only susceptible to colistin. Colistin comes from the 1940s. It's an example of a polymyxin antibiotic, and it's extremely toxic. It can lead to irreversible organ damage. It's never even been subject to a clinical trial because it's so old. It was just introduced into practice, and as a result, there's not even an accepted dosage regimen for this antibiotic. But you'll see that this is in many cases the last line of defense. Here's an interesting fact about that NIH breakout. I said that there were 3 transmissions from patient 1. Patient 1, it was shown through susceptibility tests that her Klebsiella responded to only 3 antibiotics. Colistin was 1. The carbapenems were not effective because it hydrolyzed the carbapenems. Tigicil, which is a tetracycline that I'll introduce in a few slides, and an aminoglycoside, which also is toxic, causes kidney failure in cases. What's interesting about those 3 transmission events, patient 2 and the subsequent patients all developed colistin resistance during their therapy. So we are in the precipice. You'll see colistin numbers subsequently. Here's a paper that came out in 2017. It reported observations for bacterial infections at the largest university hospital in Sao Paulo, where about 5 years, I'm gathering from the paper somewhere around 2010, 2011, they stopped using carbapenems as the frontline therapy for gram-negative bacteria because they were seeing so much resistance. And what did they switch to? They switched to colistin. So for a 5-year period, colistin was their frontline defense, and here are the resistance rates in Klebsiella pneumoniae. 2010, 74% of the infections they saw were resistant to colistin, and by 2014 it was over 90%. What's interesting, just how fast this occurs. It's estimated that in 2010, 97% of Klebsiella strains were susceptible to colistin. So just in a span of 5 years, things can change dramatically. There's a news program in the United States called 60 Minutes. Just about 3 weeks ago, they aired a special on the rise of superbugs, and they asked the question, could they become a bigger killer than cancer? And for this piece, they went to New Delhi. This is an infant in a neonatal unit in New Delhi. This baby is being given colistin. It's the only antibiotic that is effective against the bacterium that this baby has. It's estimated now that more than 60,000 babies in India die from drug-resistant bacterial infections. The same reporter then goes to, also in New Delhi, goes to a pharmacy stand on a busy street and proceeds to purchase ciprofloxacin, amoxicillin, and colistin without a prescription. And most frightening is they then visit a poultry farm in New Delhi, and you see them carrying big 50-pound sacks of chicken feed. And this is the label on that sack of chicken feed. It contains erythromycin, metronidazole, the aminoglycoside neomycin and colistin. So they're actually feeding this to, they're cultivating resistance in their community. This product is called Uprise, and I think it should be renamed Surprise because it's. But you know there is a gene that was identified first in 2015, and where was it identified? In pigs in China that were fed colistin-containing animal feed. And that was the first incidence of this gene, 2015. Now, more than 40 countries in all 5 continents have reported incidence of the MCR1 gene. In certain countries in Europe, the range now is between 28 and 43%. The World Health Organization has issued guidance on which infections we need to be most concerned about, the infections that we need to generate the next classes of antibiotics for. And they are, the top 12 or so are all gram-negative infections. And you'll see every one of these says either CR or 3G-CR. That means that it contains a gene that hydrolyzes carbapenems or third-generation cephalosporins. The top bug on the list is Acinetobacter baumannii, another gram-negative infection. And this is carbapenemase resistant. I saw a paper last week that they did a study that showed that in Turkey now, more than 90% of Acinetobacter baumannii infections are carbapenem resistant. I also want to highlight this gram-positive organism. It's the top gram-positive organism on the list, Enterococcus faecium, because the third little story I'm going to tell this evening we believe is a candidate to treat that infection. It is an incredibly lethal infection as well. It frequently will colonize, for example, a heart valve and cause endocarditis. So how have we gotten in this situation? Well, it's evolution. And evolution assisted by our misuse of the antibiotics that we have for prescribing them for non-bacterial infections, for putting them in animal feeds, and so on. The other thing that has happened is that large pharmaceutical companies, one by one, have bowed out of the fight. This is actually one of the most concerning things. You know, in the 1980s, there were hundreds, probably close to 1,000 PhD level chemists working constantly to make new antibiotics, and we were having as many as a dozen or more new antibiotics launched every year. That has stopped, and what has happened instead is that the burden for discovery of new antibiotics has fallen on the shoulders of small biotech companies, and this is a disaster because these companies are going bankrupt. So it's all has to do with money. This is an economic problem. It's not, in the current climate, it's not possible to charge enough money for a new antibiotic that it can be profitable, certainly not compared to any other type of drug treating, for example, cancer. Okay, so now that I've depressed you and frightened you, let me try and shine a little light on the problem, because I think it is an addressable problem, and I want to spend the next several minutes telling you what our strategy is. So we are incredibly fortunate that we have a large number of natural products that have evolved over millennia, perhaps 1 billion years of evolution. Chemical warfare in the soils and waters wherever microorganisms dwell. And these natural products have given us two things. They have given us lead matter. They have not given us drugs. I will make the statement that natural products are generally terrible drugs. And I couldn't back this up with example after example. Colistin, for example, which causes irreversible organ failure, is an example of a natural product that became a drug. Chemists have, without exception, been able to improve on these targets, largely through semi-synthesis, but this evening I'm going to argue that we should be pursuing fully synthetic solutions. The other thing that natural products have given us is they have identified the targets that are important, in my opinion. That would be the bacterial ribosome, which is the 3 stories that I'm going to quickly tell this evening, all target the bacterial ribosome. In addition, they are known to tackle the cell wall and, to a lesser extent, DNA. But I'm going to turn this around and say that because everybody, it seems, wants new targets. They want new classes of antibiotics, and they want new targets. If you identify a target, you wake up in the middle of the night and say, I've identified a gene in the E. coli genome, and if we can just develop a small molecule to inhibit that gene, we'll have a new antibiotic. If nature isn't already targeting that gene, then you're probably making a mistake by looking at that new target. This is a lot of evolution, and any Achilles heels have been revealed, in my opinion. The bacterial ribosome, of course, is one of the most important molecular machines in all forms of life. It is largely composed of RNA with buttressing proteins, and it is the machine that translates nucleic acid information in the form of ribosomal RNA into functional proteins. These are the 2 halves of the bacterial ribosome, and through pioneering efforts in X-ray crystallography, we now know where all of these natural products which target the bacterial ribosome, we know where they bind. And so this evening I'm going to talk about tetracycline, I'm going to talk about macrolides, and I'm going to talk about lincosamides. We also have a project on aminoglycoside synthesis, but I'm not going to talk about this evening. So I think if this isn't sending a message about the importance of the bacterial ribosome as a target for bacterial inhibition, I'm not sure what is. So here are 6 examples of natural products that target the ribosome. And one of the things you can quickly assess is that these molecules look nothing like one another, by and large. And they have a density of stereochemistry and complex functionality, polar functionality, that makes them unlike any other drugs treating non-bacterial diseases. It's well appreciated that the physicochemical properties of antibiotics are outliers. They're very different than typical medicines. And so we began our journey studying tetracyclines in the mid-1990s when I was still at Caltech. It was a problem that we would work on continuously for a span of about, well, actually we're still working on it, so a long time. And one thing that I'd like to point out is that at the time that we started this, I never anticipated that we would develop a route that would actually be scaled to make thousands of kilograms of a drug substance. I frankly didn't think that. If you had asked me, I would have been honest and said I don't think that's possible. And I think that's generally true about most of the molecules on this slide. And I am now am of the other opinion, having repeated successes. And I believe that modern chemical synthesis, properly applied, is capable of addressing these medicines. And the point is that if you do develop a practical component-based, Jakob alluded to convergency, if you have a highly convergent practical synthesis, the number of structures, antibiotic candidates that you can evaluate is orders of magnitude larger than you can evaluate through semi-synthesis. What is semi-synthesis? It's where you take a natural product like penicillin G, you use an enzyme to hydrolyze the amide bond here, and then you acylate that amine with a different side chain. That has given us dozens and dozens of new penicillins. Amoxicillin, shown here, has an expanded spectrum versus penicillin G, which is a narrow spectrum antibiotic. In the case of oxytetracycline, chemists at Pfizer learned how to chemically remove that hydroxyl group, giving an acid-stable tetracycline. This molecule undergoes dehydration in the stomach of a human being and forms a toxic byproduct. Similarly, erythromycin is acid sensitive. Chemists at a small company called Pliva in Croatia developed a 4-step synthesis of azithromycin from erythromycin involving a Beckmann rearrangement, which expanded the ring from 14 to 15. This is azithromycin, a Pfizer drug now, and one of the most successful antibiotics of all time. Introduction of that basic nitrogen had huge consequences in terms of PK. It persisted in the body for days after administration. You'll see in data in this lecture that azithromycin has resistance to azithromycin is now quite widespread. And that and other reasons, I would caution against taking this antibiotic. The cephalosporins are all made by semisynthesis, just through manipulations at 2 positions, through acylation of 7-amino cephalosporinic acid and this 3' position. The first cephalosporin was launched by Eli Lilly in 1964 and was a gram-positive antibiotic only. It was not effective against gram-negative bacteria. But over 50 years, additional 2nd, 3rd, 4th, and 5th generation cephalosporins were launched, largely through introduction of polar functionality. And these drugs became broad-spectrum antibiotics. And the sweet spot is, I'm told, are 3rd generation cephalosporins, which now there's widespread resistance. These are broad-spectrum antibiotics, and for a time were frontline antibiotics for gram-negative infections. I think one point I'd like to make this evening is that if you're into antibiotics discovery, you have to take a long view of things. It's easy to imagine somebody in 1980 saying, you know, I've just spent the last 20 years doing semisynthesis of cephalosporins. That's an old class of antibiotics. We need to move on to new targets and new classes. And those arguments would be difficult to refute. And yet then we would have missed 3rd, 4th, and 5th generation cephalosporins. So modular chemical synthesis is a path forward. This is identifying building blocks represented by colored spheres here, which can be assembled in a highly convergent coupling. So you'll see this illustrated several times, but it's when you take 2 complex molecules, halves if you will, ideally halves, and you bring them together in a way that forms sometimes more than a single bond and multiple stereocenters. And the more of those convergent couplings that you can do, and the more you can minimize linear steps, that is A going to B, going to C, going to D, the more efficient your route will be. And then by modification of these building blocks as well as the intermediates, you can literally explode and very easily have access to tens of thousands of new compounds. And then briefly, I want to mention again that all carbapenem antibiotics are fully synthetic. There is a factory or factories that manufacture nothing but carbapenems. Because they're allergenic, it's not possible to make other drugs at these plants. So Merck invested millions and millions of dollars to develop the process chemistry to make imipenem and then later ertapenem. And I am told that during the 1970s, there was an extensive period of time when virtually the entire Merck Process Research Group, which was one of the best in the world, was dedicated to manufacture of carbapenems. The initial route was 14 steps, largely linear. It was expensive. I'm not even sure they were making a profit initially, but then subsequent process improvements brought the costs down, and they, in fact, Merck has turned a very good profit on carbapenems for many years. You know, the carbapenems have been off patent for many years, and there's not a single generic carbapenem antibiotic on the market. Why do you think that is? It's because it's too complicated to synthesize, and they can't meet the purity standards for new drugs. So there's actually a bonus if you have a synthetically complicated drug. You may never have to face competition or be concerned about generics. One of the things, though, that the reason that I show this slide and I talk about it is that I like to challenge people and say that if as a society we are not willing to make this kind of investment, and I can tell you large pharma is no longer, you would never see this happen at Merck today. If we're not willing to make that type of investment, then we're going to have to suffer the consequences. Okay. So up until last year, all tetracycline antibiotics were also made by semi-synthesis, by decoration of fermentation products. Once this hydroxyl group was removed, acid-stable molecules could then undergo nitration, which allowed introduction of dimethylamino group here and the generic antibiotic minocycline. A group at American Cyanamid, led by the late Frank Talley, developed tigecycline, where they introduced a second nitrogen atom at position 9 and decorated it with this very interesting and unusual T-butylaminoglycine. I knew Frank Talley. I had the great privilege to know him. He was a big supporter of our tetracycline synthesis effort, and ironically, in a cruel twist of fate, he died due to endocarditis, a bacterial infection in his heart. Tigecycline is the tetracycline that I mentioned that patient 1 was susceptible to. So this drug was effective at treating that Klebsiella infection. So in the mid-1990s, following actually a successful ene-diyne project, dynemicin A, I was struck with the idea that we might tackle tetracyclines in a convergent way by assembling the C ring. So from an AB precursor and a D ring precursor, we felt that we could invent chemistry that would allow us to bring those two entities together. What I didn't know at the time is that it would take us about 10 years before we identified the structure of the AB enone and chemistry to prepare that molecule. But once in hand, Dr. Christian Lerner very quickly identified an anionic cyclization reaction. We had to invent this, where we can deprotonate in the benzylic position, and that anion then carries out a Michael addition, a highly diastereoselective selective Michael addition, and an enolate intermediate that is formed then snaps shut onto this phenylester to give, essentially as a single diastereomer in high yield, this substance shown. This was a very fortunate event because this stereochemistry corresponded to biologically active tetracyclines. And what's particularly neat about this is that once you have the AB enone, as we call it, in hand, you can access a fully synthetic tetracycline in just 3 steps: cyclization and then 2 deprotection steps. And the chemistry was general. Dr. Q. Wang showed that with this different D-ring precursor, she could prepare minocycline. in 3 steps. But what was more important is that we could access tetracyclines that could never be accessed by semi-synthesis from natural products. For example, this pyridine containing D-ring was accessible and prepared by Jason Brubaker. So at this point, it was pretty clear that this AB-enone was a very important molecule. And our initial synthetic route to that required 10 steps and benzoic acid as a starting material. And it allowed us to make a few grams, enough that we could make various tetracyclines and test them. But it was evident, I had a postdoctoral coworker who spent a good 6 months trying to scale that route, and there were serious problems. And so Jason Brubaker, as a first-year graduate student, took on the challenge of developing a second synthetic route to the AB-enone, and this one was much more convergent. So we have this furfural derivative and this isoxazole component, and these, we don't purchase these have to be made. They're now outsourced, I believe, in China, but they can be brought together and in just 5 steps transformed into the AB-enone. This forms the outline of the route that Tetraphase Pharmaceuticals scaled to the level of thousands of kilos with a lot of process innovation. This is a photograph of the first kilos that were produced. It's one of my favorite pictures. And this is how we present the synthetic route. So this is the furfural, and this is the isoxazole. This is the convergent coupling. Then we do 4 linear steps, and then we introduce the D-ring precursor, and this is our AB-enone and 3 steps. So in 2006, I believe Tetraphase Pharmaceuticals was founded with the sole goal of commercializing this technology. And while they were developing the process chemistry to scale the AB-enone, they began to hire medicinal chemists and make tetracyclines. And within, by early 2008, they had identified a molecule, TP434, which went on to become eravacycline. And that was identified within the first 400 molecules. The last I knew, they had eclipsed 3,000 fully synthetic tetracyclines. Most of these had antibacterial activity, and most of them could not have been made by semi-synthesis. It was very fortunate that they identified eravacycline early on, because otherwise the company probably would not have made it. They almost didn't make it anyway. You might remember that in 2008 we had a rather large financial crisis, and there was a time when I worried that the company would fold. But because they had this compound that showed such promising activity, they were able to move forward through into a phase 2 clinical trial. This is a vial from that phase 2 clinical trial. There are 50 milligrams of powder in the bottom of this vial. This is eravacycline. And I show this slide because it reminds me to tell you that it costs more to put this powder in a sterile vial than it does to make the powder. In other words, the cost of goods is not a factor here. This is an affordable drug substance. This is a scalable chemical synthesis. Now, eravacycline looks a lot like Tigicil. It is the first fluorocycline, so there's a fluorine atom rather than a dimethylamino group at position 7, and there's a different glycine side chain at position 9. Those might seem like relatively simple changes. First, I'd say that introduction of a fluorine semi-synthetically would be incredibly challenging. But in terms of a patient receiving this drug, they're vastly different drugs. Why do I say that? Well, it turns out that there are issues with Tigicil. In fact, I think in 2014 the FDA issued a black box warning against the use of Tigicil because they were seeing patient deaths. Why? Because doctors, recognizing that Tigicil was sort of uniquely effective against some of these very frightening infections, were starting to use it for some of the worst infections out there, things like hospital-acquired pneumonia or ventilator-acquired pneumonia. And the problem is that Tigicil has got a dose-limiting toxicity. It is very poorly tolerated, and it causes nausea and vomiting in patients. And so as a result, they can't give enough of the antibiotic to cover those really frightening pathogens, and as a result, they're treatment failures. This molecule, for whatever reason, has a much better safety profile. It can be dosed higher, and as you'll see, it's 2 to 8 times more potent, and it has better pharmacokinetics than Tigicil. And when you sum across or multiply across those 3 factors, in terms of efficacy in patients, this is a vastly different molecule, I would assert. So, eravacycline, Xerava, was approved in the United States and in Europe in October of 2018, and they started, it was approved before that. They started selling it in October. Sales are anemic. They've just been a full quarter of sales. It was something like $340,000 of sales. And you can buy a share of Tetraphase stock for about 85 cents last I checked. There's a problem. I want to show you some of the data for eravacycline. This is off of Tetraphase webpage, excuse me. So this is an evaluation of 4,000 patient isolates that were known to be multi-drug resistant, gram-negative infections from New York City. And what they are measuring here is an MIC90. MIC90 is defined as follows. It is the dose of a given drug that you have to give to treat 90% of the infections you're looking at. So for example, here's our friend, drug-resistant Klebsiella pneumoniae. And so if we look in the back row at ceftriaxone, one of those third-generation cephalosporins, you can see it doesn't work anymore, not in New York City. Meropenem, a carbapenem, the MIC90 looks to my eye to be above 4 or about 4, so it's pretty borderline. But you can see that with eravacycline, we're looking at a number of, I believe it's less than 2 micrograms per mil. Tigicil is just back here. So eravacycline is effective against all of these infections, and it's one of the types of antibiotics that we need nowadays, and yet it's not profitable. So in the 10 years while eravacycline was trudging through the clinical trials, I had a lot of time to think about next steps, and to be honest, with some considerable trepidation, I was thinking about tackling macrolide antibiotics. Macrolides and tetracyclines share parallel histories. They were both discovered at about the same time. Erythromycin was discovered by Eli Lilly scientists who scooped up a little sample of dirt on a visit to the Philippines. Just like tetracycline, erythromycin is unstable in the acidic environment of the stomach and forms this bicyclic ketal, and this compound causes motility issues. And I've taken this drug, and I can tell you it is an awful, awful drug. In large measure, to avoid that side reaction from occurring, chemists in Japan worked out a 6-step synthesis of clarithromycin, where they just place a methyl group on this hydroxyl group, and that prevents that ketalization. Then I've already talked about azithromycin. You can imagine it's incredibly challenging. Look at all the hydroxyl groups. There must be 7 or 8 of them in this molecule. How do you functionalize one selectively, say a tertiary alcohol? Very challenging chemistry. The latest members of the macrolide antibiotics have a sad history, frankly. Telithromycin was introduced in 2004, and shortly after it began to be used in a widespread way, they began to see rare but fatal forms of liver toxicity. And so that was withdrawn from the market. And the Congress brought the head of the FDA before them and said, how could you have allowed this poisonous medication to advance into society? And so thereafter, the FDA became much more hard-nosed about clinical trials for antibiotics, which is one of the reasons that which hastened the exit of large pharma from pursuing antibiotics. Solithromycin, I'm afraid, is unlikely to be approved as well. This is the latest semi-synthetic macrolide. It's made in 16 linear steps from erythromycin, and now you'll see this diagrammatically. And in their phase 3 clinical trials for community-acquired bacterial pneumonia, patients had elevated levels of liver enzymes, and for that reason, I think it's unlikely to be approved. So I was greatly motivated to tackle a problem this challenging by the arrival of postdoctoral student Dr. Ian Seipel and then graduate student Xian Zhang. They were the pioneers in this project. They were the ones that were willing to roll up their sleeves and consider developing a practical synthesis of macrolides. It almost sounds oxymoronic, but they did it. And what they showed was that for one particular scaffold, these 8 building blocks, which in our lab were all prepared in amounts from 30 to 100 grams, could be combined in a highly convergent way to assemble macrolides quickly. And with the foundation of macrolide pharmaceuticals and other small biotech that was designed to commercialize this technology, each of these building blocks was synthesized in amounts between 5 and 6 kilograms. So this is how those building blocks are assembled. In light of time, I'm not going to drag you through all of the chemistry. I will show you one reaction. But to make a kilogram of a final macrolide, fully synthetic macrolide, you would have to start with less than a kilogram of most building blocks, a little bit more than this building block. So we start with a convergent coupling, followed by a linear step, another linear step. And here we begin with a convergent coupling, another convergent coupling, and so on. The overall yield from the beginning to the end is 43%. And it's true whether you begin with these starting materials or these. That's a coincidence. This, by contrast, is the linear synthetic route from erythromycin to make 1 kilogram of solithromycin if we assign a yield of 85%. Those yields are not made public. So you'd need more than 12 kilograms of erythromycin. This is a different scaffold using slightly different starting materials here. The others are the same. And this is a truly unique scaffold, and it's a biologically active scaffold. So we have a 14-membered ring, which erythromycin has, but we've replaced the carbonyl at 12 o'clock here with a nitrogen atom. So hopefully having the improved PK of azithromycin, but a different scaffold. The reactions that brought these building blocks together in some cases had to be invented. I'll show you one here. This is an aldol reaction. It's a derivative of glycine, and you'll notice that the amino group of glycine is not protected. And it's an amide with a molecule known as pseudoephenamine, and we've shown that this is a superior chiral auxiliary versus pseudoephedrine and has the desirable feature that it can't be transformed into an illicit drug, and so it's safe to use, and it's very easy to make on kilo scale, as they did at Macrolide. This glycine derivative is deprotonated with 2 equivalents of lithium hexamethyl disilazide, and that combines with this chiral methyl ketone to give in 98% yield a single diastereomer. So this forms an alpha amino beta hydroxycarbonyl compound, and that functional group motif is very common in antibiotics. You'll see this reaction again in a few slides. That is a truly remarkable reaction, and it is a general reaction. So at Macrolide Pharmaceuticals, along with continued research in my lab, we've now made close to 2,000 fully synthetic macrolide antibiotics. And a goal was, I didn't mention this, but macrolide antibiotics are not effective in gram-negative bacteria. They don't get past, or they don't stay within. If they do make it past, they're subject to efflux. There's a paper by O'Shea and Moser. It's a famous paper in the field. I encourage you to have a look at it, where they make the point that most antibiotics that target gram-negative bacteria tend to have molecular weights between 200 and about 650, and they tend to be very polar molecules within this box. Macrolides, by contrast, are up here. They're too big and they're too lipophilic. But with chemistry, we changed that. So we moved the macrolides from here to this area of space. And when we did that, we found that we had multiple scaffolds, different scaffolds, that were effective against gram-negative bacteria. So I want to show you a little data, and I apologize for not showing structures. But these are about a half dozen clinical isolates of E. coli and Klebsiella, and the resistance genotypes are indicated here. ESBL stands for extended spectra beta-lactamase. NDM stands for Nudeli metallobeta-lactamase, another lactam hydrolyzing enzyme. And first you'll see that none of the other macrolides are effective at all. We knew this before: erythromycin, clarithromycin, azithromycin, telithromycin. They don't get in. And then here are other antibiotics. We have ceftazidime, a third-generation cephalosporin, a carbapenem, meropenem. It's about flip a coin whether that works. Fluoroquinolone, ineffective. The aminoglycoside amikacin is largely effective, with some notable resistance. But as I mentioned, aminoglycosides have issues with renal toxicity. But here are 3 different scaffolds that we prepared that are all different from one another. And this is where we first cracked gram-negative activity. And look at the numbers. It's 2 to 4 micrograms per mil. Probably not potent enough to move forward as a drug substance, but once you breach the wall, look how they're broadly effective. They don't, these bacteria have never seen macrolides, so they've never evolved resistance mechanisms, not yet. With continued synthesis, we were able to improve those scaffolds. And here's Klebsiella pneumoniae, multi-drug resistant. We have an MIC of 1 microgram per mil. So this is a remarkable achievement to transform a class of gram-positive facing antibiotics to bona fide gram-negative antibiotics. We have a few candidates that are moving forward as clinical candidates. But I have to be honest with you and say that I'm concerned about the likelihood that these molecules will ever see the light of day. There's really a crisis out there. Wall Street is not supporting antibacterial research. Lastly, I want to tell you about some completely unpublished data. It concerns work that has occurred exclusively in my laboratory towards the Lincosamide antibiotics. The first molecule natural product identified at Upjohn in the 1960s was lincomycin. It's a very interesting molecule. You can see that it's readily dissolved into two components by hydrolysis of this amide bond. There is this interesting 8-carbon sugar with an amino side chain and a chiral secondary alcohol, and then a methyl thio group at the anomeric position. And then the other component is known as 4-propylhydric acid. They were pretty lucky at Upjohn. They did one semi-synthetic step. They transformed that secondary alcohol into this chlorine modified structure with inversion of configuration, and that's clindamycin. That is an approved drug and has been approved for several decades. Clindamycin is useful, was useful. It treats gram-positive bacterial infections, and it's a nice alternative to beta-lactams. For example, if you have a patient that is allergic to beta-lactams. It has 100% oral bioavailability. It is effective against syphilis. It also has anti-malarial effects. And there is widespread resistance to clindamycin now, a type of resistance known as MLSB. I'll talk about that in a slide. But the biggest issue with clindamycin and why it's really not used nowadays is that it promotes Clostridium difficile. C. diff, as it's known, is a horrible infection that occurs, ironically, as a consequence of taking antibiotics. And the worst antibiotic that you can take to promote C. diff is clindamycin, and we'll see why that is tonight. What happens is that clindamycin we have C. diff in our guts right now, but we don't suffer from C. difficile because it's kept in check because it's competing with a large number of competitors. When you take an antibiotic like clindamycin, you kill its competitors, but you don't kill C. diff. And so it overgrows and it sporulates. And so in the worst case situations, you have to have your colon removed. I buy clothes from this guy, this shop in Boston, and a couple months back, I've known him for years, and he said, "So how's the research going?" And I said, "Oh, we're really excited about this new antibiotic that we found in the lab. It doesn't promote C. diff." And he said, "Tell me about it." He said, yeah, that he had to have his colon removed and now uses a bag. Very, very serious infection. Okay, so we know through the beautiful work of the late Tom Stites, Nobel Prize-winning work, that clindamycin spans, here, you can see it here through a slice of the bacterial ribosome. It sits between the two subunits. And PTC is the peptidyl transferase center. So this is where the two tips of the two tRNAs meet and actually form the amide bond of the newly growing protein. And then this is the peptide exit channel where the protein is expelled into the environment. And you can see that this little molecular wrench is preventing the transfer of the synthesized protein through the exit channel. And so as a result, Lincosamides are bacteriostatic and they freeze protein synthesis. That can actually be a very good thing because many of those bacterial proteins cause toxic shock syndrome. And so instantly freezing bacterial protein synthesis can be quite a good thing. A little bit of a close-up shows that there are many different specific interactions. The protonated amino group of this propylhydric acid is actually replacing a scaffolding divalent magnesium cation. And then there are lipophilic interactions here. And the peptide the peptidyl transferase center is about right here, and we'll see this in a movie in a minute. In addition, there are a series of hydrogen bonds, very specific hydrogen bonds with RNA base residues and the hydroxyl groups of this elaborated sugar. And so as a result, you cannot modify these 3 hydroxyl groups. Now what's interesting is this is sometimes called a pseudo-base pair interaction. There are 2 hydrogen bonds with this adenosine residue, adenosine 2058. It turns out that one of that MLSB resistance mechanism that I talked about codes for a protein that goes in and methylates that amino group. It places 1 and 2 methyl groups to transform NH2 to dimethylamino here. And you can imagine what that would do. Not only would it disrupt that hydrogen bond, but it would create a steric interaction. And as a result, methylated ribosomes no longer bind clindamycin, are not inhibited by clindamycin. So an interesting question you might ask yourself is, could we perhaps overcome that type of resistance by picking up other interactions elsewhere in the molecule? And I can tell you the answer is yes. So I'm doing a terrible disservice to Dr. Matthew Mitchell-Tree. This is about half of his PhD thesis research on one slide, but Matt developed a fully synthetic route to the amino sugar, one which allowed us to modify substituents at position 1 and this side chain. He had to invent some powerful chemistry to do that. He was able to, using a literature catalyst, was able to carry out a diastereoselective nitroaldol reaction, and then he discovered fortuitously that when he subjected that product to triethylamine, the nitronate would open up the epoxide and so set this stereotriad here. He then removed the acetylenic silyl protecting group, put on another silyl group selectively on this secondary alcohol, removed the N-oxide with trimethylphosphite, and then reduced the carbon-nitrogen bond in a directed reduction with sodium triacetoxyborohydride. So that sets up all the stereochemistry along the periphery. And then cyclization with a tungsten catalyst gave this glycal, which could be epoxidized, and then with stereo retention, nucleophiles, be they carbon or sulfur, could be introduced at C1. I told you you'd see this reaction again. This is now being used to synthesize a wholly novel lower scaffolding element, a much more elaborate structure than the propyl hydric acid. And it began, as Matt showed, through another bond pair forming reaction that he had discovered, where the glycine aldol reaction, as before, gives the syn alpha amino beta hydroxy amide, which then undergoes cyclization upon warming to form this elaborated proline residue. He later introduced an allyl group and carried out an olefin metathesis to make this oxapenoproline motif. And so I just want to identify that olefinic carbon is C7 prime. Why did we target this, and why is C7 prime of interest? If you overlay that molecule onto clindamycin, you can see that substituents on C7 prime would project into a lipophilic pocket which projects where the amino-acyl bond is being made. Dr. Catherine Silvestri, in her PhD thesis work, showed that she could carry out a regioselective Wacker oxidation to place a ketone at this position, and then through a series of 3 non-selective steps, was able to introduce various side chain residues here. We learned that this is the desired stereochemistry at that position, gives rise to more potent antibiotics. And this particular compound with this isobutyl side chain emerged through MIC testing as an especially exciting antibiotic candidate. So much so that we decided to move forward into murine infection models. And I'm fortunate to have Dr. Amory Pisapati is a postdoctoral coworker in my lab who is not only adept at the microbiology of MIC measurements, but he is also an expert in rodent infection models. And so we're doing these experiments in my lab. We could not do those experiments with the discovery synthetic route. This is what led us to identify the molecule, but a team of 3 postdocs developed a second generation route that has permitted us to access the larger amounts of material that we need to support animal studies and I hope perhaps, if we're fortunate, human studies one day. Again, we are using 2 components that are somewhat elaborate but made in just 4 or 5 steps. This enol triflate, which is made from 3-hydroxyproline, a very cheap starting material. And then using a pseudoephonamine-directed alkylation reaction, we arrive at this enantiopure iodide. These are then coupled. We form an organozinc reagent, and then a palladium-catalyzed coupling gives rise to this molecule. Now, this is very much a work in progress. It's provided the material that has allowed us to be in mice now, but the yields are not all worked out. Some of these steps are in process. For example, this intramolecular directed hydrosilylation reaction only proceeds in yields of 40 to 50%. Now, that has to be optimized. With this in hand, we then carry out a transketalization reaction in hexafluoroisopropyl alcohol. That's a rather bizarre solvent. That was necessary because in the next step we do an ionic hydrogenation, and if we use something like methanol, we observed some reduction of this carbon-oxygen bond. But with this better leaving group, we get exclusively reduction to make the oxapane. And so you can see our stereochemistry here was bought in the pseudoephonamine alkylation, and then the other steps are the same. So this is an X-ray crystal structure of that molecule bound to the bacterial ribosome. It's a movie made by Yuri Polikhanov, our collaborator, wonderful collaborator. This is the peptide exit channel, and there is our antibiotic. And you can see the 2 tRNAs right here in green and blue. And this will come into better resolution in a second. So there's the A site and there's the peptidyl transferase. And you can see that isobutyl group is effectively wedged between those. And you can see why this immediately shuts down protein synthesis. And there's a substantial post-antibiotic effect, meaning that when you remove the antibiotic, protein synthesis remains stalled for a considerable period of time. I just want to close very quickly with a little bit of data. I want you to see that this has truly exciting activity. These are staph strains, many of them clinical isolates, all of these resistant to multiple antibiotics. You can see that none of these strains is susceptible to clindamycin, and azithromycin is even worse. And yet we're effective. You know, MICs are reduced against the methylated ribosomes, but quite impressive nevertheless. I have to say this strain I have a lot of experience with in other classes of antibiotics, and it is one of the worst strains that we have. This MIC of 4 is actually very impressive. So you can see that with this methylated ribosome, we're still binding and we're still inhibiting. This is streptococcal activity. And again, these C-ERM-B, that means that it is constitutively methylated. So it is a methylase gene that is always turned on. And that renders bacteria resistant to not only clindamycin, but also erythromycin. And the MIC goes up a bit, but at 0.25 micrograms per mil, that's quite a fine number. And then here is the organism that might cause endocarditis. And I think that might be, in terms of greatest unmet medical need, I think that might be a direction one could pursue with as a path to the clinic with this agent. And these numbers are really quite impressive. And you can see that the other, this is vancomycin. So these are mostly vancomycin-resistant strains. And then lastly, and very importantly, this is Clostridium difficile, and you can see that clindamycin has an MIC of 8 micrograms per mil. So on the wrong side of inefficacy. And this is another commensal that we use. It's not important for this evening's lecture. But you can see why clindamycin would promote overgrowth of C. difficile, because it doesn't kill it, but it does effectively kill its competition. We have an MIC of 0.25 micrograms per mil. So our expectation is that we will not promote C. difficile overgrowth. There is a guinea pig model to assess this, and it is something that we will pursue after we've demonstrated an ability to cure infections in mice. So with that, I'm done with the chemistry portion. I've tried to acknowledge students on the slides. I'm talking about chemistry over a span of, 25 years. So it's challenging. I'll call out Christian Lerner, who did the first AB plus D cyclization, and Jason Brubaker, who developed the synthesis of the AB enone that is now scaled. This is Ian Seipel, and this is Zhang Zhang. They were the courageous souls that initiated the Macrolide synthesis program. And I stole this slide from Matt Mitchelltree from his thesis talk, and of course he doesn't have his own picture, he has my picture, so I apologize for this. I have to get a picture of Matt. This is Dr. Amarnath Pisipati, who's doing our animal studies, Catherine Ioana, and an undergraduate, Jack Stevenson. So I'm very much indebted to them and to you for the opportunity to be here this evening. I'd be happy to take any questions.
So the floor is open to questions, and please don't hesitate. I'm sure Andy will be happy to discuss.
Thank you for your presentation. With the fast evolving nature of the superbugs, do you think hunting for the new antibiotics is the only solution, or do we have some alternative?
That's a great question, and I tell you, you quickly get me outside of my range of expertise, but I'm aware of things. Vaccines. In theory, vaccines could be developed and are being developed for certain types of bacterial infections, and so I think were those vaccines developed, and if people actually took the vaccines, that is a potential solution. One hears about bacteriophage. In fact, I believe that bacteriophage have been used in the Soviet Union for a few decades. I'm frankly not an expert in bacteriophage, and I can't assess how that compares as a strategy versus synthetic antibacterial agents. But you know, I guess I didn't really sum up my end point argument, which is these old classes of antibiotics can give us many new. We need chemists to be synthesizing, and you know, I think there's way too much emphasis on classes in when talking about antibacterials. It's almost like I use a term, molecular racism. If I showed you the activities and I didn't show you the structures, would you care? If it's safe and effective against the infections you care about, would you care if I told you that, oh, that's a Lincosamide antibiotic? You shouldn't, but a lot of people do.
Question. When you test your compounds, do you also test them for the ability to create resistance? And if so, at what point?
Not immediately, but eventually you always do that before you get very far. Mm-hmm. It's a pretty easy experiment to do. Mm-hmm.
So is it like repeated or continuous exposure of
yes,
bacteria to the
exactly.
compound?
And what you look for is you measure. If you are seeing resistance rates of about 1 in 10 to the 7th, then you're probably not going to have an antibiotic. But if you're about 1 in 10 to the 9th, then you have a good chance. That's my understanding. Okay. And it's one of the first things you do before you would ever advance to a clinical situation.
Yeah. Okay. So how I'm wondering, you know, how often have you had a case when the compound was really potent against the bacteria, but then it created resistance very quickly?
It's not happened to me yet. Mm-hmm. We certainly, the tetracyclines, they looked at that, and there was very low acquisition of resistance. And the biologists at Macrolide have done that with, I believe they've done that with our most advanced candidates. But we've not attempted yet to, maybe Amory did and didn't tell me about it. He's always tinkering in the biology lab, but I don't think we've yet tried to do that.
Great. Thanks a lot.
Why are new antibiotics not profitable? I was told years ago that, and it may not be true anymore, that there is still usually or always some combination or some old antibiotic that will work in a situation. It's just finding it. Is that still the reason why new antibiotics can't find a market?
It's a complicated question, and there are multiple facets to it. I'll try and give my best answer. So one is they can't pricing. You know, you can be prescribed a cancer medication that they might charge you $100,000 or $200,000 American dollars for, and it might prolong your life by a few months, but it doesn't cure you of your cancer. You can have a bacterial infection such as the Klebsiella that I've talked about, and you can die in 2 days, or you can take a 2-week course of eravacycline and be cured, but you can't charge $50,000 for that. You certainly can't charge $100,000. So there's this disparity in pricing, but it's worse than that. Even when new antibiotics are out there, there is great resistance among infectious disease doctors to prescribe them. They prefer to save them for when they really need them. It's my impression that in cases they would rather go with an existing cheap antibiotic that they know that in the past has worked, but might not work in this case, and have a failure and then go to a plan B rather than to prescribe the new antibiotic. So there's a reluctance, there's a slowness to adoption. You know, you can plot antibiotic sales over time, and the curve always looks like this. It's a straight line, and it takes years for sales to ramp up. And if you're a small antibiotics company with a burn rate of $100 million a year, you don't have years for sales to ramp up. It's fundamentally an economic problem, and I'm sad to say that the solution is going to have to come from governments, the U.S. government and the European Union. I'm not very sanguine about the US government right now.
Sorry, so I guess my question relates to what you just mentioned, which is that the solution would have to come from government because you have a really interesting experience of science and business, I suppose. Do you have any communication with sort of legislative bodies to try and address this? Do you know people who have any communication?
What was the last part you said?
With legislative bodies, the government or anything?
No, I don't, but I know people that do. For example, there is a company, not a company, there is an organization that is funded in part by the Wellcome Trust and also by entities within the United States. It's called CARB-X. And they have a pool of some millions of dollars that they dole out to small antibiotics companies to promote research. And the head of that is a law professor at Boston University. I apologize. His first name is Kevin, I think it's Uderson. And he lobbies Congress. And he told me that there was a bill before the last legislature, but that it did not pass. So I'll tell you what a solution might look like. If you are a small antibiotics company and you successfully bring a bona fide, useful antibiotic treating the infections we care about, if you bring it to market, you take it all the way through clinical trials, then I, the US government, will give you a voucher that you can use to extend the patent life of any drug that you wish in your portfolio by, say, 2 years. And you make that voucher transferable. So you might not have any other drugs in your portfolio, and you say, well, this is useless to me, but you could sell it to Pfizer or to Merck, and they might pay you $500 million for it because they could extend the patent life of Lipitor or some billion-dollar drug. That would work overnight. I can almost guarantee it. And it would work in two ways. Not only would it drive all this income into small companies, but all the major pharmaceutical companies would reopen their investigations into antibacterials, because why should they pay $500 million for it when they can start up their own research activity and earn that credit themselves? The problem with that is if you are a congressman or a senator in the United States that votes for that, it's going to be seen by the public as you've just taken a step to enrich pharmaceutical companies. You've allowed them to sell their drugs at an inflated price for another 2 years. We're going to vote you out of office. Does it make sense what I'm saying? It's a very, very complicated situation. You know, the scientific aspects of it are hard enough. I think I can handle some of those, but the financial, the economic, the political consequences, it's a mess. It's a great question you ask, and biofilms are a concern. I have to confess that I'm not an expert. Like I should know the efficacy of eravacycline in a biofilm situation, but I apologize, I don't know. It's definitely something to be concerned about. I know a little bit about China. I know that eravacycline is now, it's not approved in China. It's in phase 3 clinical trials there. Eravacycline has been through 2 or 3 phase 3 clinical trials for complicated intra-abdominal infections, so it's going to work. And it will be approved in China. I am not aware of discovery efforts in China. It's a great question because They're very, very clever people, and they know how to solve big problems. Sometimes there are advantages to having a somewhat authoritarian regime, but I don't know what their commitment is.
There's one more here. Paul, just.
Yes. Yeah, it's actually one of my postdocs. Actually made me aware of that transformation. Let's see, let's go one more. I'm sorry I went over that so quickly. So I don't know if I'm answering your question, but this is the transformation. So you pre-treat with this, it looks like hexamethyl disilazine. This is a silyl transfer reagent. So what that does is place a dimethyl silane group on there. So there's an oxygen silicon hydride. And then Karsted's catalyst is a very complicated platinum catalyst, which brings about the catalytic transformation. It adds a hydrogen here and a silicon there. And the important thing is it's a cis addition. So it comes from the top face because of stereochemistry here. And then you carry out an oxidation, a Tamau-Fleming oxidation with hydrogen peroxide. And that sets this center and this center. Did that answer your question or no? Exactly. And there aren't, the other way to do that would be a hydroboration, except the hydroboration goes on the other face. And we haven't given up with hydroboration because you can put a boron hydride on that oxygen, but we've never been able to get it to add. I just before I left, I asked a student to heat it up above 100 degrees because I think at some point it has to go, but it hasn't yet. The yield of this is there's a side product that we haven't figured out how to diminish yet, but I'm confident that with continued work we will. I'm sorry, I'm having trouble understanding your question.
There was like a increase of the scale of the reactions. So there was a photo that your people were performing the synthesis in like 1 kilo scale per bottle, right? And I'm asking, it was still performed in the glass or in the still reactor?
Yeah, we only use glass. And there are very few cases where we've gone up to 1 kilo. I'm pretty safety conscious these days, and with large scale comes large danger. Just working with a large volume of solvent I'm always nervous when a student is doing a scale up, so we try and keep things reasonable, but we work only in glass. You're welcome.
All right, one more.
I would like to ask you, is there any chance how to avoid development of resistance?
How to avoid resistance? Yeah, actually, I'm glad you asked that. Again, I'm straying outside of my area of expertise, but one of the things that we can do is just to use our antibiotics wisely. And there were 2 countries in the European Union. I think Denmark was one, and perhaps the Netherlands was the other. I might be telling you the wrong countries. And they adopted a system where they restricted, let's just say carbapenems. I don't know which ones, but let's say they restricted carbapenems for only the most serious patients in hospitals, and they absolutely didn't use those in the community. And so they tiered their antibiotics. And one of the things that they saw, which is a little bit heartening, is that when you remove the evolutionary pressure, when you don't put colistin in your animal feed, bacteria will lose these resistance mechanisms. Most of these resistance mechanisms come at an evolutionary cost. You can methylate your ribosome and overcome resistance to erythromycin or lincomycin, but that methylated ribosome might not function as well as the normal ribosome. And so when you remove the pressure of the drugs, if you don't put them in the community, then the resistance mechanisms can recede. So I think we have to be very smart about how we use our antibiotics. But you see the conundrum there, that if you're saying, you've tetraphase, you've just developed eravacycline, and you're able to treat this Klebsiella pneumoniae, but now you're saying, well, we're not going to prescribe that very much. We're going to hold that in reserve for the sickest patients. Then the company goes broke. So it's the economic model is broken.
And do you think that it's possible to approach this problem also from like more molecular view, let's say to target some different pocket on ribosome, or to choose different therapeutical target?
That is a brilliant question, I think, and I absolutely do think so. And in fact, the proof is already out there. I didn't talk about it, but there is a class of antibiotics. It's often said the only new class in the last 30 years. They're called oxazolidinones. And the founding member was linezolid. And linezolid binds in the ribosome. It's near where these other antibiotics binds, but it's different. And linezolid is a very effective antibiotic, very important antibiotic for many years. And I think that in the future, if you were to say, I have an idea about hitting this binding pocket on the ribosome, and maybe computation will get to the point where it could actually tell us which molecules to synthesize, I think there's very great possibilities. I think there's a very bright future for targeting different or slightly different areas of the bacterial ribosome in the future.
Thank you.